basically we have the assumption that tests performed for the events A, B, C, and D were accurate, robust, and sufficient. I'll be going into that in a moment, but actually that assumption is widely contested. We also have the assumption that assessing the traits or the novel components in this case, that is all that needs to be assessed. So basically you would look, for example, just at the Bt protein. You don't need to look at many other components of the plants, you look at the particular protein or the trait. Well, that also is contested. Then the next assumption is that there is no interaction between the 8 different genes, their pathways, their proteins, their traits, or between the genes and indeed the plant's own genes. Again, that is an assumption that once was held quite highly and ranked, but more and more scientific data is coming that actually says we also have to put away with that assumption. It's heavily contested. The next one is that synergistic, cumulative, or other combinatorial effects do not exist. That means that the interaction actually does not give rise to risks and harm. Again, that is very widely contested. So already now, that's quite interesting, already now one should actually say in the scientific community there is no consensus that doing risk assessment only for the original 4 plants should be sufficient. It's rather the opposite. The consensus is much more that we need to really look at the new event as a new event when you put it together, because all these assumptions here are flawed. For example, we have SmartStax also currently going through the European system. That is one of the comments of the Austrian Federal Department for Health, which notes that in the dossier, the notifier says that the safety of all the trans proteins have been discussed in detail in other applications for authorization. That's what the notifier says. Now, this concerns, amongst other things, the history of safe use, structural description, and digestion in simulated gastric fluid. Well, in contrast to this, the Austrian Department for Health actually points out that there is no history of safe use of the new recombinant proteins, but also concerning all the Bt toxins, the history of safe use cannot be argued on the basis of the safety of Bt sprays applied in organic farming, because that's what it's looking at as a reference. The inserted genes, for example, in the plant are truncated and arranged with expression modulating DNA parts, which is quite different to that what you find in the bacteria. So therefore, if you get the data from and your assessments from proteins that are expressed by bacteria rather than those from plants, you cannot actually compare it properly. That is basically what this is saying, but it also states that some of the proteins here have not even been assessed in plants in the European system yet. Basically establishing structural and functional equivalence of these test proteins and the plant-derived proteins adds uncertainties to the interpretation of animal tests. So even the animal tests which have been done, we cannot rely upon, which means we need to have new ones. Which is quite interesting that we also had it recently in the European Union that one of the genetically modified events was actually withdrawn because the company had been asked to, in this case Monsanto, had been asked to provide more information on animal feeding, which actually uses the closest parental isoline as the comparator, because that is another issue. If you have a genetically modified plant and feed that to the animal, and then you feed any other corn as a control, then you cannot compare properly what the changes have been. So basically what one needs to look at is the closest isoline, which is the parental line, before genetic modification has taken place. If you don't do that, then it's very difficult to do the comparisons and to have a data that actually is valid. In this case, the company had not used the isoline and therefore was asked by the European Union to please provide feeding data on that, upon which the company withdrew the whole application. So that shows it's not just like which proteins we test, but also like what do we compare it to in terms of the baseline against which we have to judge is something altered, is something changed, is it toxic or allergenic. And in the ARTEC group, which Lucy mentioned, actually there's quite a number of discussions to which extent one might have to go back to, because with the gene stacking, what is the comparator? Because you start, if it's 8 genes, the immediate parental lines would already be transgenic, would be GM lines. Can they be counted as controls or as the comparator? And the European Union and quite a number of us in the ARTEC group say, like, no, that is not valid. And so there's discussions to be had. But of course one can look at them, but that should also, the comparison should also always include the non-transgenic lines. Well, I'll be looking at now some of the unintended and unexpected effects, where one thinks like, oh, we know that gene, and that is fine, it's not a problem, so maybe one has it assessed in one genetic background, but not in the other. But things like, if they are closely related, that should be fine. In this case, the gene for the alpha amylase was taken from the bean, or from a bean, a common bean, and then genetically modified into, or genetically engineered into a pea. And actually the results, when tested in feeding trials, showed that this protein now, although it had the same DNA sequence and the same amino acid sequence, it appears that actually now it had an altered structure and an altered immunogenicity. It needs to be looked further at, whether it's glycosylation or other modifications which have taken place. That all of a sudden a protein that otherwise was completely safe was immunogenic, and on top of that actually it made other components in the seed, in the storage proteins of the seed, allergenic. So that is a data which was produced in Australia by the Hogan Group. Actually, I'll show you a slide here about the BT corn, which was tested here by Saxena and Stotsky, or by Pershman et al. And you can see that you have a BT corn, and all of a sudden when you look at it, you have an increased lignin component in it. And lignin is what you find in wood, what makes wood like hard, and actually it helps to build the cell wall and the structure, which then enables plants also to transport fluids. And it's also very good as a protectant against herbivores, sort of pests. And in this case, you all of a sudden have an increase by 33 to 97%, or by 18 to 28%. The question still is, and we don't understand it, what does the addition of an insecticidal protein like the BT, how come that the plant is now reacting with producing extra lignin? And this is just to indicate there are changes that one does not expect, that one does not that are not intended, but they will occur. So if you just look at the trait, you might miss them.